Hello, and a very warm welcome to everyone joining us for today's Select Science webinar, Improving Analytical Method Performance Through Quality Risk Management and a Design of Experiments Approach. My name is Sarah Thomas, and I will be moderating today's presentation. I'm delighted to be joined today by two speakers, Joseph Turpin, Director of Chromatography Products and Services at S-Matrix Corporation, and Tran Pham, Business Development Manager at Waters Corporation. Today, Tran and Joseph will present an overview of quality by design approaches to analytical method development, known as AQBD, the regulatory views on this approach, examples of design of experiments in AQBD, and case studies demonstrating software-assisted AQBD method development. After the presentations, we'll move on to our question and answer session. So please feel free to ask any questions for the Q&A session at any time during the webinar, and you can do this by clicking on the tab to the left of your screen. So without further delay, I would like to hand over to Tran and Joseph for today's presentation, and I'd like to thank them for presenting to us today. Please go ahead.
Hello, everyone, and welcome to today's Select Science webinar. My name is Tran Pham from Waters Corporation, and today's webinar will be on improving analytical method performance through quality risk management and a design of experiments approach. We will start off by explaining a few words on AQBD, followed by a case study presented by Joseph Turpin from S-Matrix. Today's webinar will cover the following topics. We will start off by briefly describing analytical quality by design, also referred to as AQBD, followed by the current thoughts from supporting regulatory agencies. We will then move into defining and providing examples of design of experiments and highlighting its benefits. We conclude the webinar by presenting a case study that utilizes a software-assisted design of experiments for method development. AQBD stems from applying quality by design processes from ICH Q8 to analytical methods. The definition of AQBD is defined as a science-based risk management approach to developing robust methods by identifying critical method parameters through risk assessment. Once critical method parameters are identified, design of experiments take place to determine whether changing one or more of the parameters impact method performance. The definition of design of experiments can be summarized by a process of setting up a series of tests to determine how inputs affect the outputs. More specifically to analytical method development, design of experiments are used to find operable instrument ranges and to identify sources of variability and the effects when varied. Here are common terms typically used when referring to AQBD, referenced by Phil Borman from GSK. Terms used in this webinar are summarized by the following. Analytical target profile is a document that states the requirements of the method. The method must meet these requirements to ensure fit for purpose. Critical method parameter is an analytical parameter that has a critical impact on method performance when varied. And lastly, method proven acceptable range is an allowable range a parameter can change within, and the change does not affect method performance. Currently, the ICH is writing 2 new chapters highlighting the importance of applying AQBD to method development. More specifically, ICH Q14 will be dedicated to proposing a harmonized way for method development. The main goal of this chapter will be to provide guidance on how to increase efficiency and robustness when developing methods and when making post-approval changes to them. The ICH is committed to the importance of AQBD and intends to publish these chapters within the next 2 years. This is the definition of QBD found in the current ICH Q8 revision for drug manufacturing. This definition is also applicable to analytical methods. Successful method development requires a systematic approach and should start with predefining method objectives. Predefining method objectives provides a clear path to test critical method parameters and leads to better method understanding when varied. In addition, understanding and establishing control strategies to prevent varying parameters ensure good method performance. So how does AQBD for methods fit in with QBD for pharmaceutical drugs? A drug's critical quality attributes are requirements that must meet a set criteria in order to confirm the drug was made for its intended purpose. AQBD is introduced when analytical methods are required to test and generate critical quality attribute results. AQBD ties back into QBD when control strategies are established for the method. Not only will establishing control strategies ensure method performance, the method results reinforce the manufacturing controls used to ensure high-quality product. There are many ways to set up design of experiments to test critical method parameters, and here are some examples. One factor at a time, OFAT, is still commonly used as a design of experiment. For example, when making changes to a monograph, one factor at a time can be used to increase the separation efficiency by increasing the column length over particle size ratio. One factor at a time can also be used to determine limit of detection and limit of quantitation by varying the injection volume when developing new methods. The major drawback of using one factor at a time is it limits the ability to fully understand how different factors interact and affect results. Full factorial leads to a more comprehensive understanding of how interacting factors affect method performance. And lastly, to have full understanding and the ability to predict method performance when parameters are varied, a design of experiment using modeling software can be used. As you increase the complexity of design of experiments, you increase the understanding of what is needed to ensure method robustness. AQBD is an integral part of a new approach to method management called Method Lifecycle Management, MLCM. An AQBD method development approach sets a higher rate of success for method validation in stage 2 and method performance in a routine environment in stage 3. The 5 key AQBD method development steps are: define the method's objectives and expectations by creating an analytical target profile. Perform risk assessments to determine critical method parameters that affect method performance. Design and execute experiments with applicable DOE to test the effects of varying parameters. Gather knowledge from the results of DOE or perform more experiments to implement control strategies to reduce method variability and ensure method performance. Major pharmaceutical companies are adopting AQBD method development procedures and have experienced high success rates. Hovione is a global contract manufacturing organization located in Lisbon, Portugal. Since incorporating AQBD to their method development procedures, they have seen a drastic increase in method robustness, reliability, and a decrease in method failures. Now we are going to show you a case study demonstrating the benefits of AQBD via a case study. The case study will be presented by Joseph Turpin from S-Matrix. Take it on, Joe.
Thank you, Tran. I'll be discussing the case study of a modernization of an existing monograph method from the USP of a major API. When we were approached with taking a look at this method, we were tasked with developing an organic impurities method that was more modern, that would better separate the impurities and reduce the need for multiple methods. The existing monograph utilizes multiple methods to determine drug substance impurities, either because of the lack of detection or coelution of the impurities. It also doesn't adequately separate degradation products. Current method is also an isocratic method, and as you can see from the slide, there are several problems with this, including coelution of multiple impurities. There was also at the same time that we were approached a proposed EP monograph method that was being considered as a method to harmonize with the USP. And we were asked to take a look at the robustness issues of this method and possibly do some experiments around the robustness. When we took a look at this method, and with our approach to the method itself, these were the goals that we had in mind for our target method. And typically, kind of the typical parameters that you would expect in trying to design a method. So retention between 1 and 20 for K prime. Our target for the API was greater than equal to 2 for resolution of that peak, and all other peaks, if at all possible, greater than or equal to 2 for the resolution, with no individual result less than 1.5 or baseline resolution. We also wanted tailing to be less than or equal to 1.5, We wanted this to be a single-step gradient, which was one of the issues with the proposed EP monograph method, in that it had 3 very shallow gradients right around the center of the API elution. We also wanted a method that was above ambient, so we could transfer this to a lot of different temperature zones and not need any kind of an ion pairing agent. When we assessed the proposed EP monograph method that the USP was looking at harmonizing, we noticed 3 primary parameters that we felt would be a problem without even any experimentation. And that was the oven temperature of 20 degrees. The method also had an ion pairing agent, and as I mentioned earlier, had a multi-step gradient, which had 3 steps right around the vicinity of the API. In addition, very few of the impurities next to the API separated very well. So these, just looking at the method from an evaluation standpoint, these method parameters pose significant risk to being able to transfer the method. As Tran mentioned earlier, DOE, or design of experiments, is a widely recognized method of doing development, particularly in the process industry. And if you consider HPLC as a process, then you can directly transfer this approach. And when looking at DOE, when you start to look at the numbers of variables that you may be using in experimentation, it looks like a tremendous amount of work. But statistical sampling, using a lot of the statistical models that are in wide use, shows that you can reduce that quite a bit through statistical sampling. So, for example, when we consider 2 variables with 5 study levels each, on the left of the graph here, you would see there would be possible 25 combinations of methods that you would have to run. Using a design of experiments approach with statistical sampling, you can reduce that to 13 methods. An important piece here is that what you're also trying to do when you do these statistical methods is to fill in the gaps without having to actually run the experiments in between. When you start to look at adding 3rd and 4th factors to the study, particularly when you add different types of factors, which are represented here as in column type, then you're moving out of the numerical scale. And numerical factors are factors which are used at levels represented by numbers on a continuous scale, and that has meaning, and for which intermediate levels between the defined levels also have meaning. This is the case for pH, since the numerical scale has a meaning. Increasing levels, 2, 3, 4, etc., are related to an additional continuous property of the factor. Also, if one defines these study levels such as 2, 3, and 4, then intermediate levels such as 2.5 or 3.5 also have meaning on the continuous scale. Categorical factors are things like column type, buffer type, and they represent a disjoint or continuous system. Even if one defines the level as numbers, such as column 1, 2, or 3, or operators 1 and 2 or 3, numbers don't exist on a meaningful scale. Operator 2 doesn't represent twice as much of the property as operator 1. For example, you can't have column 1 and 1.5 of column 1. So those are represented as non-numerical levels of column type. This is the basis of our screening experiments. So this is an example to illustrate the statistical efficiency of a DOE design. If you had 5 levels of gradient time, 5 levels of pH, 4 levels of column type, you'd have 100 possible combinations. In a screening design, and the designs we present through the Fusion QBD software, you would have 30 runs plus 6 repeats, which would give you approximately a 3-fold efficiency increase in the experiments. This slide shows our typical approach in a screening study. And typically what you do in the screening study is prepare your experiment as wide as possible that makes sense. So for example, we would look at pH across a range from 2.7 to 5, for example, in 1 pH unit increments. We would not do a range across the entire range of pH at larger units. Gradient time depends on the column, But in this particular study, we set this gradient time between 10 and 25 minutes, and we're running our profile of our organic from 2 to 95%. And the column types, we picked 4, and these 4 columns are selected with regard to polarity differences, hydrophobicity, and a good source to look for the differences in the chart type would be to look on the water selectivity chart that's available on the WATERS website. To keep our system initially at a very good response, we looked at acetonitrile as a solvent. If you were to look at combinations of solvents, that would be done in, could be done in a screening, but it would be an either or. So you'd have a situation that would be solvent type in that case. Pump flow rate, 0.4 mils a minute. We're running a 2.1 by 100 millimeter by 1.7 micron column. So an average flow to keep from overpressuring across the entire range of columns, pHs, and conditions. We set a flow rate that we felt and tested to be sure that we wouldn't overpressure the system. And we're preparing our buffers online by mixing a stronger buffer systems, 20 millimolar formic acid and 20 millimolar ammonium formate. This design is created with these actual experimental variables by the user, and they're translated into Empower methods as ready-to-run sample sets. When one is doing screening or even doing any initial development on methods, you typically have lots of different types of chromatography that's occurring, and so you have a variety of chromatograms that can occur. And so this is an example of a very ugly chromatogram where you actually have 8 compounds here, and you can see we're co-eluting several, and peak shape is bad, and so on. So the question asked is, how do we track these invisible peaks? And typically when you're doing development, you want to track the peaks so that you know the identity and what is co-eluting. With our approach here, we actually do what we call trend responses. And they're basically some selectivity trends on the chromatography. We're looking at things that the chromatographer typically looks at in a somewhat of a pictorial fashion or graphical fashion, but we turn those into quantifiable parameters by setting these what we call trend responses, which are things like the number of separated peaks, how well do the peaks separate, do you get baseline separation, do you get better than baseline separation, what's the tailing like? There are several parameters that you can pull in from the Empower result set. Anything available there can be pulled in, so you can pull in things like tailing, peak height, etc. And we also automatically identify those things, so we do a peak count on the number of peaks well resolved, and we identify the max peak by the area response as well. So we can automatically calculate the resolution of the max peak, which might be your API. An example of a trend response that we would use to measure the quality of the separated peaks would be, an example is the number of separated peaks. So in this example, we show 2 chromatograms from a BEHC18 column at 2 different gradient times, both at pH 2.7. And in the top chromatogram, you see 6 peaks separated, and the bottom you see 8. So you can see that just moving to a longer gradient in this case would improve the separation slightly. We look at another example trend response. The number of baseline resolved peaks starts to tell us how well peaks are separated and gives us a quantifiable measure of the peak separation. So in this example, we look at the 2 different gradients again at the same pH, but 2 different columns as well. And as you can see in the bottom chromatogram, we've started to improve the separation, even though we still have some problems with peak elution times. We have improved the peaks and now see several peaks that are separating above baseline resolution. We present the searches based in both mathematical and graphical modes, and depicted here is the graphical representation. The areas that are shaded are areas where we fail to meet the criteria that we set. So for example, The orange area would be the where we don't meet the first peak retention time that we seek. The unshaded area represents the area where we meet all of our goals simultaneously for all of these parameters that we set. Each successive response is plotted, and so we end up with an overlay that shows where we have the possibility in our screening experiment of being able to achieve our goals. Now, what we do pick in this case, even though our center point in this method shows a 25-minute gradient time at pH 4 on the CSH phenylhexyl, to promote this to an optimization, we pick a slightly larger area because these are all mean responses and they're much wider variations of the parameters. So we want to dial into this area, including areas that fail, so that we see the areas of failure on the optimization method. An additional thing we do when we look at this, either in a screening run or an optimization run, is to experimentally validate the methods. Not talking about validation itself, but a validation of the experimental results. And so we look internally and occasionally externally at the data acquired and integrated with Empower. In this example, there are 2 data points that were collected by our experiment that are within the area to be explored with optimization experiments. The desired experimental conditions lie between these 2 points, as you can probably see in the peaks around the API or following the API. You see obviously the small peak moving back and forth between the API and the next impurity behind it. So we suspect that the optimum experimental conditions would lie somewhere between or around these conditions. We also make sure that our method and our data are consistent by doing an external assessment where we exported the set of conditions determined to be the best as a confirmation to be run with Empower. As you can see here with this chromatogram, there's potential, but there's still work that needs to be done to improve the critical peaks. If you look at what's highlighted in the blue box, you can see that there is fair separation around the peaks, but we could improve peak shape, obviously, and probably could also improve the separation. So to begin method optimization, we take that graphic that we looked at earlier and the conditions that we set, and we expand that region to include the areas where we may show failure as well. We're moving now away from just the things that are most likely to affect selectivity, which are pH, gradient, slope, and column type. And then we will now add in things like flow rate and temperature, which are typically secondary effectors of separation. This also allows us to calculate robustness for those parameters as well. We've changed our gradient profile to start at 1% organic, which from indication of the initial screening, we were still having problems with the retention of the first peak. So if we drop the organic down a little bit, we'll see if that has an effect. And to shorten the method, instead of going from 2 to 95%, we're maintaining the slope and changing that from 1 to 50% because we saw that there were no peaks eluding past the 50% mark, and then adjusted the gradient time to give us that same slope. And as you saw from the graphic earlier, we selected the CSH phenylhexyl as the best type. Strong solvent type is still acetonitrile, and we'll be looking at 20 millimolar ammonium formate at the pH ranges of 3.6 to 4.2. We're now down into 0.3 unit increments and working more to get more granular data in a narrower space. Data are processed in Empower with consideration for consistency after your runs are complete. So you would do processing with a processing method, and if necessary, name your components and then identify each of the components in Empower. Those then can be imported into Fusion QBD, and then we do the tracking and monitoring of all the parameters that you're interested in that are available in the Empower result set. If you have access to the Waters Acuity QDA mass detector, we now employ a new tool called Peak Tracker. After the data are processed With Empower, with the consideration of producing consistent integration of real peaks, the data were imported into Fusion QBD. The new peak tracker tool was used to identify the peaks by assigning the names to the associated masses acquired by Empower and import. So there's no need to name the components in Empower in the processing method. The tree on the left shows the individual experiments, and if you select a run number on the left, it displays the imported UV and total ion chromatogram. After identifying the peaks in a representative run, all peaks for all the runs are identified by their mass and associated names. Additionally, the peaks are identified on the UV chromatogram, and the location of non-absorbing peak is denoted in the UV chromatogram, allowing the non-absorbing peak to be placed in the chromatogram in a simulated fashion. And in the boxes above, we highlight the area where the component called H, the letter is placed in the chromatogram above just to show its placement. And then you also see it below, directly below, where it has been detected, ionized and detected on the QDA. The boxes below are where you assign the names of the components. And when the data come in from Empower, they are matched to their mass. So if masses are available for your compounds, you can quickly identify those and then identify across all of the runs without having to track the peaks in Empower. This graph displays the average performance goals we set for resolution and retention of the first peak. The unshaded region displays the conditions where all of the mean performance goals are met. For example, where no peak falls below resolution 2. The shaded regions represent areas where we don't meet our expected goals. And in this case, we're looking at an overall resolution map in the blue, and the red is the retention of the first peak. So on average, if we work in this region here, our method will meet its goals. This graphic shows a new feature that we've added, which is the resolution map display. And the red area is where the method meets all of its performance goals. And if you place your cursor at the crosshair and move that around, you'll see an actual simulated chromatogram with the results of our expected chromatogram. This graph shows the resolution map in a 3D fashion. And as you can see, as we showed in the graph in the earlier page, where we centered our crosshairs in the center of that, you can see partly why we did that, in that there's a very nice flat region in the kind of a tabletop on the resolution map in the red area. Note also that nothing falls below resolution of 3 in that area. So none of our critical peaks, and that would be taking every peak and every possible critical resolution pair in our entire experimental space and ensuring that we don't fall below our goal of 2 in our resolution. You'll notice that on the right of the chart that it does fall off fairly quickly, kind of in a waterfall type fashion. But if you look at the entire range, we still stay above resolution of 2 all the way down to the bottom of this chart. But to remain stable and in a mean performance approach, we still want to push this to upper levels. The previous graph shows the average performance of the method. To consider method robustness, we must consider also the variability of the method. A well-accepted approach for measuring the performance of a process employs metrics used widely in the chemical and pharma industries to gauge processes and set specifications. This is called process capability. If the variability of the method at plus or minus 3 sigma is completely coincident with the lower and upper spec limits, the capability index is 1.0. If one sets the specification plus or minus 4 sigma and the ratio of the spec limit to difference to the variability is 4/3 or 1.33, this is considered in the industry to be a capable process. Our next step was to employ the internal Monte Carlo simulation in the software to calculate the variability of the method from the data we already collected, and to calculate the process capability of the method for each critical pair at values we set to reflect what we would expect to encounter in labs around the world, not just running it on the system that we have in-house. The center point of the method then in this graphic situates the method at pH 3.8 with a 14-minute gradient, 0.45 mils per minute flow, and 45 degrees, as you can see in the graph. Now, the graph also depicts the area in the unshaded region where you can operate this method in a robust fashion. So anywhere within that unshaded area, you will meet all performance and robustness goals. Now, you'll notice that we have this center point, rather than sitting in the very center, which would still also work. We also brought it down, and it's closer to the resolution of the first peak, but you can see the process capability is set to where we still would be robust within the region. If we look at the graphic of mean performance versus robust performance here, with the resolution maps displayed in a heat map and a 3D map, you can show that if we sit in the middle of the region, then we're sitting in a very stable region for any of the critical pairs. And in this case, we're looking at the API, a degradant peak, and the E peak in our chromatogram. This also shows that no pair in this very stable region falls below resolution of 3. And here is the final method that we arrived at, and our performance criteria on this. You can see that we have no peak that's tailing above 1.5. All peaks are well resolved. We're using a single gradient. The K prime that we found on this method was greater than 1 for the first peak and less than or equal to 15 for all remaining peaks. And the resolution between the API and any of the neighboring peaks is greater than 3. As part of the robustness strategy, we also look at limits in terms of what we would expect if we transferred this method somewhere, and the critical parameters that we've identified from the method itself from actual experimental data. And we set these limits on flow, pH, gradient time, and temperature. So flow can vary plus or minus 0.05 mils per minute, pH plus or minus 0.15. The gradient time we calculate as a 2% compositional change using the data from our gradient time. And then temperature was less of a variable, but can be varied plus or minus 3 degrees. We determined these parameters as the critical parameters for a robust method. And an example, you see that pH is critical in maintaining the separation of peaks following the API. As the pH changes, it affects that resolution, and that peak actually moves between the API and the peak following it. We also notice that there are several factors that interact, and this demonstrates the need to look at your method development in a multivariate approach, because with a one factor at a time approach, you never discover the interaction effects, which can be linear and nonlinear. And in our example, we determined that pH and flow rate, as well as gradient time and flow rate, were factors that also had interaction effects. And this is why you can't use slope alone to determine global optimum. So in summary, I hope we've demonstrated to you that our initial evaluation of a proposed method indicated the need for a more robust, modernized method, and that by applying analytical quality by design and a multi-factor software-assisted DOE approach to improve method performance, identified the critical method parameters, multiple parameter interactions that comprise method robustness, and that they're tested and identified and can be controlled. Hopefully also we demonstrated to you that method performance prediction is achievable and that you can actually drive your methods to the goals you have developed. Also that method development screening and optimization efficiencies are increased and development time is reduced. And in both of these cases, the screening and the optimization experiments were each performed in overnight runs in an automated, unattended fashion. And last but not least, that non-statisticians can use statistical tools without the need to being statisticians themselves. And within Fusion QBD, you can employ statistical method development in an efficient fashion.
Thank you very much, Tran and Joseph, for your interesting presentations. So it's now time to move on to our question and answer session. So if you haven't asked your question yet, then you can still do this to the left of your screen. So our first question here is, what was the system used in the case study?
So this is Tran. The total system used was the Waters H-Class Plus. PDA, QDA are the two detectors, and the software used was Empower, CDS, and Fusion QBD that also has Peak Tracker.
Great, thank you, Tran. Our next question here is, does AQBD only apply to chromatographic methods?
No, it doesn't. Basically, any procedure that requires an analytical method to be robust and provide accurate, reliable data to determine product quality of any kind can benefit from AQBD approaches.
Great. The next question we have, developing methods using AQBD approaches seems to require more work and longer development times. How can the added time and labor be justified?
Developing analytical methods using AQBD is perceived to require more time and more work compared to traditional methodologies when first implemented. However, once a company has implemented AQBD procedures unique to their analytical development and workflow, the benefits of AQBD does come in more quickly in realizing the benefits over reducing operating costs and providing higher confidence is much appreciated and respected for the workflow.
Thanks, Tran. The next question here, did you apply the Fusion software for the full process? Is Peak Tracker part of the Fusion software?
This is Joe. Yes, Fusion QBD was applied through the entire process, from the screening through the optimization, and really the benefit of applying that all throughout is a more robust method and the full automation of the tools available through Empower and Fusion QBD. And Peak Tracker is part of Fusion. It's a new feature in a new release of the Fusion QBD software.
Great, thanks, Joseph. The next question here is, do you use Empower to do your DOE studies?
We use Empower to perform all of the chromatographic experiments. The designs are all done within the Fusion QBD software and exported directly to Empower as ready to run sample sets. And then data is processed and then imported back into Fusion QBD for this statistical analysis.
Great. The next question, how do you know how to set the gradient slope values and stop time values when running the screening stage?
The important things to consider, several things to consider here, are the limitations of your system. So mobile phase miscibility, column pressure limitations, etc. What you really want to do is set as wide a gradient as you can so you retain peaks in the early part of the chromatogram and elute all of your peaks within the gradient, if at all possible. So typically just run, for the screening stage, run a long gradient, and as wide a gradient and as long as a slope as possible, with a couple of different slopes.
Great. And the next question here. Please can you share what you know of clients submitting AQBD-based method development in their regulatory file and how the regulatory agency respond? Did the client company receive regulatory relief in allowing post-approval method changes as long as the changes were within the operating space?
We don't receive all of the information about products that have been submitted from our clients, but a couple of clients have mentioned that they have submitted methods and submitted with the statement that they developed the robustness during method development and not during traditional validation. And so far, those have been accepted, and they haven't received any questions about the method robustness development during the development process.
Thanks, Joseph. Remember, if you have any questions, you can submit them to the left of your screen. And we have another question here. Are robustness control limits experiment results or statistically derived values?
Combination of the two. We do derive those controls statistically from all of your data, and we use a Monte Carlo simulation process to calculate all the variability of the data that you already generated. So there's no additional work that you do to get those. For establishing those, the important consideration here would be not just look at the specifications of the instrument you're running, but look at specifications of instruments that you might transfer that method to anywhere around the world. So you can widen your specifications and your controls and test the method and see if it's robust under those conditions. So really the answer to the question is a combination of the two. They're statistically derived, but you want to also widen those to consider what you're going to do with the method.
Thank you, Joseph. So that's all we have time for today. Thank you very much to Joseph and Tran for today's informative discussion and presentation. So if you've asked a question and we haven't answered it yet, then we can follow up with those offline. And thank you to everyone joining us online and for your interesting questions. If you do have any more questions, please feel free to email me at editor@selectscience.net and I will follow up with your questions for Tran and Joseph. Remember, you can also download a certificate of attendance and a related application note in the Resources tab to the left of your screen. And if you'd like to listen to today's webinar again or to invite a friend to listen, it will be available to watch on demand in a few days' time. So goodbye and thank you once again for joining us.